Good afternoon, everyone. Am I audible?
Yes, ma'am.
Okay. So, welcome to the week 3 live interaction session for the course Classics in Neuroscience. Let me start by discussing some of the previous year assignment questions, followed by addressing any doubts that you might have. And we can discuss some of the concepts associated with the week 3 lectures. Is my screen visible?
Yes, ma'am.
Okay. So, first question. Who proposed the term synapse to describe the special contact between nerve cells in 1897? So, let me go to the course content. Yes. So, initially in the 18, sorry, hmm. initially, uh, Ramoni Cajal, who is considered the father of modern neuroscience, when he was looking at the the tissues under the microscope, he had proposed the neuron doctrine. So he said that neurons are individual entities that interact with each other through some contacts between their axons and dendrites. So after this, when there was a big debate about the whether the um, whether the neurons whether it's a neurons are separate entities that interact with each other or whether they are a continuous reticulum. So then there was a big debate about that. But slowly later, when people started doing further experiments. People slowly came to realize that after several years of controversy, people came to realize that neurons are indeed individual cells that interact with each other. So, just a minute. So, during that time, Charles Sherrington, later Sir Charles Sherrington, who received the Nobel Prize, so he was an English physiologist, and he was actually trying to understand how the spinal cord reflex functions work. Just a minute. I have some presentation that I can show you. Yeah. So initially, Cajal, when he had observed the mammalian cerebellum under the microscope, this is a diagram that Cajal made. So at that time, Cajal had noticed that. So this is regarded as one of the first, like. Observations of the synapse by this drawing by Cajal. So he had observed that there are certain connections or contacts that are made by by the in the nervous system, and they are so the nerve cells are basically discontinuous or independent elements, and he had said that there are some contacts that might exist. So now later, when Charles Sherrington was studying the reflex action in the spinal cord, he said that these the sensory nerves and the motor nerves, when they interact with each other in the spinal cord, so basically this region, He said that these contacts that exist, these he said that he decided to name them as synapse. So synapse is actually, it derives from the Greek term synapsis. And from that, Sherrington decided that he will call these contacts between the axons of one neuron with the dendrite of the other as synapse. So this is a quote from Sherrington, in which he says that this the tip of the twig of the arborescence, basically the axon, the tip of the axon, basically is uh, not continuous with the dendrite. So they are not continuous with each other. They are separate from each other, but they contact each other. So they are two separate entities, the axon of one nerve cell and the dendrite of the other. And this point of contact between the axon and the dendrite, he decided to call this special connection as synapse. So that is why the answer to the first question is that it was Charles Sherrington who proposed the term synapse to describe the special contact between nerve cells in 1897. And remember, he did this in 1897 and not 1987. So remember when, like remember that this was a very early discovery in the late 19th century. Next, who made the groundbreaking discovery of the mitochondria's fine structures in the early 1950s? So this was done by George Ballard. So when George Ballard and Sanford Paley, the two of them were studying the structure of, I mean, studying the nervous system using the electron microscope. That is when George Ballard made this observation. Let me just go to that section. Yes. So George Ballard and Sanford Paley were electron microscopy was just booming at that time. The use of electron microscopy had just started, and some new advances in the staining and tissue sectioning were also had also just been made. So Sanford Paley was at that time working at Yale. So Pallard and Paley, the two of them, they were collaborating with each other. And Pallard had actually run away from Romania at that time because of the Nazi rule in Romania. Pallard had basically run away, had sought refuge. So Pallard and Paley, the two of them, used electron microscopy, and they tried to understand the structure of the synapses. I tried to understand the structure of the neuronal, the organelles of the neurons and so on. So that is when, in 1954, the American Association of Anatomists, Pallard presented his work, which was on basically regarding the different structures that were present within the neuronal cells. Remember, before this, light microscopy was in use. Before electron microscopy came up as a technique that could be used, it was only light microscopy that existed. And the resolution of the light microscopy was far lesser than that of electron microscope. So it was not possible to actually observe the interior, the organelles of the cells to a great degree of detail before the electron microscopy came up. So using the electron microscope, George Ballard actually observed the presence of not just mitochondria, but also several other structures within the neuronal cells, and as well as the dense plasma membranes of the neuronal cells at the synapses. So that is why the answer to this question is George Ballard. Next, which statements are true regarding the neuron doctrine? So the what is the neuron doctrine in the first place? The neuron doctrine states that neurons are actually cellular entities which are discrete in nature. So they do not have reticular connections. They are not a continuous reticulum. They are discrete cells which contact each other and and, uh, and this contact can be through synapses. Okay, so on the basis of that, which statements are true regarding the neuron doctrine? First, it was disproved by the discovery of gap junctions. No, so that is not correct because gap junctions are basically the one second. Let me go to that section. It will be much easier to explain in that way. Um, yes. So gap junctions are basically the structural basis of electrical synapses. So here you can see these are gap junctions. So basically these allow direct transmission of electrical synapses between neurons. But the presence of gap junctions does not invalidate the neuron doctrine. And why is that? That is because neurons are still discrete fundamental entities. They are individual cells that are able to form contacts with each other. So these gap junctions are also mediated by the presence of certain protein complexes, and that facilitates the transmission of electrical information. So neurons are still separate cellular entities, and these cellular entities are the basis of the functions of the nervous system. And because they are cells, they also naturally they also naturally come under the cell theory. That's why the correct options in this for this question correct answers options for the question are: which statements are true regarding the neuron doctrine? Neuron doctrine is indeed based on the cell theory because neurons are cells. Next, evolved to include complex neural circuitry. Yes, so neurons interact with each other, and the result that results in the formation of neuronal circuits. So that is why the third option is also correct. And the second option, neurons are considered the basic building blocks of nervous system. Yes, indeed, neurons are considered the basic building blocks of the nervous system. So for this question, all three of these options are correct. The first option is incorrect. Next, the electron microscope revolutionized cell biology in the early 1950s. What magnifications did it offer compared to light microscopy? So, like I said, uh, till the early, ni- I mean, uh, till the 1950s, there was only light microscope that was available for scientists to use. The advent of electron microscope, which happened in the early 1950s, offered the uh, much higher magnification of up to 50,000 times more than that of light microscope. So if you go to the text, you will find it in this part. The advent of electron microscope allowed for ultrastructural observations and magnifications up to 50,000 times or more. Yeah. So the electron microscope offered magnifications of up to 50,000 times compared to the uh, that of light microscopy. Next, which discovery or theory is correctly matched with its corresponding scientist or time period? Okay, so there's a list of discoveries that is, yeah, sorry. So there is a list of discoveries that were of these discoveries that were made. Let us just compare. So, discovery of animal electricity by Luigi Galvani came first. So he was the one who first discovered that there is some electrical impulses that exist in the uh, body. So that was something that was discovered by Luigi Galvani in 1791, right? Next, Hans Christian Ørsted discovered the electromagnetic effect in 1819. But here it is incorrectly mentioned that he studied advances in understanding the propagation of and mechanism of action potentials. So that this is incorrect. Jules Bernstein gave us theories on resting membrane potential and action potential. But here it is mentioned that he uh, discovered the use of galvanometer in electrophysiology. That is incorrect. And development of Nernst equation was not carried out by Carlo Monte, Monteucci. It was actually carried out by Walter Nernst. So therefore, the correct option is discovery of animal electricity in frog muscles by Luigi Galvani. Which of the following statements accurately describes the breakthroughs in neurophysiology involving the squid axon and the iron flow hypothesis? Okay, so the squid axon and the iron flow hypothesis was, the experiments involving the squid axon and the iron flow hypothesis were carried out by Hodgkin and Huxley. So what did they do? So Hodgkin and Huxley worked using the squid giant axon, and they just yes. So Hodgkin and Huxley had used the squid giant axon for their intricate mechanism measurements of the action potential. But for that, they required the equipments such as electronic amplifiers and oscilloscopes, which, because of advances in technology, were thankfully available at that time. And because of the availability of such techniques, they were able to observe nerve activity to the extent that they could. And this allowed them to understand the changes in membrane potential that actually constitute what is the action potential. So, the first option is correct because it says that technological advances in electronic amplifiers and oscilloscopes were crucial in enabling researchers to observe and study nerve activity, including dynamic changes in membrane potential. And Hodgkin and Huxley, because of the advances in technology and the presence in electronic amplifiers and oscilloscopes, they were actually able to carry out these experiments. So the first option is correct. Second option, Hodgkin and Huxley's iron flow hypothesis proposed that the action potential results from the selective flow of chloride and calcium ions across the nerve membrane. This is actually incorrect because Hodgkin and Huxley's iron flow hypothesis says that it is sodium and potassium ions, not chloride and calcium ions. So let me just go to the slides. Yeah. So Hodgkin and Huxley, when they were studying, they had they had seen that it is changes in the sodium and the potassium ions across the membrane, which is responsible for the propagation of the action potential. So when the yeah. Next, the iron flow hypothesis was supported by rigorous experimental evidence, fundamentally altering the scientific understanding of neural communication. Yes, this is true. It is because of the presence of such rigorous experimental evidence carried out, rigorous experiments that were carried out by Hodgkin and Huxley, that we now that this hypothesis was actually of great importance, and it greatly increased our understanding of how the action potential actually works. Next, the iron flow model explained how differences in iron concentration across the membrane lead to rapid chemical changes observed during the resting potential. This is wrong because the iron flow model explained not chemical changes but electrical changes that happen. Remember, the movement of the sodium and the potassium ions causes rapid electrical changes that are happening during the resting potential. It is not chemical changes that are happening. That is why this option is wrong and the first and the third options are correct. Next, what is the primary purpose of the Goldman-Hodgkin cards equation, GHK equation? So at that time, there was a requirement of some some principle or some equation that would help to account for why there is a such a rapid reversal of the potential. Wait, yes. So at that time, there was a requirement to understand or some way of formalizing why there is a uh, during an action potential, why there is a reversal of the potential. And for that, the 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 Goldman-Hodgkin Katz equation actually helped in formalizing how this membrane potential is arising because of the like a combination of the both the permeability as well as the concentration gradients of potassium, sodium, and chloride ions across the membrane. So the presence and the formation of this GHK equation was truly important because it helped account for the reversal potential in addition to also talking about the permeability as well as the concentration gradient of these ions across the membrane. So now keeping this in mind, we are naturally able to see that the fourth option is correct or incorrect because it says that it describe to describe the movement of ions across membranes without accounting for potential reversal. That is not true because the GHK equation it was made to account for potential reversal. So the third option says that to formalize how membrane potential arises from ion concentration and permeability gradients. That is true. So the third option is correct. Fourth option is incorrect. Next, what did Edgar Adrian attribute the regular rhythm of impulse generation to in his study of afferent impulses from receptors in the frog's sternocutaneous muscle? So Edgar Adrian was Lucas's student. So when Lucas died in a factory accident, wait, let me go to the slides. Yes. So Edgar Adrian was Keith Lucas's student who took over Keith Lucas's lab upon his death. And Edgar Adrian and his students worked to understand how nerve impulses were happening. So when so they were using these muscle spindles, basically the frog's sternocutaneous muscles, and they were trying to understand how nerve impulses were happening. And they were using the electrometer and a three-stage amplifier for this purpose. Remember, they were not at that time having any ability to record intracellularly. These were extracellular recordings, right? So when they were trying to do the recordings, they saw that there is a regular rhythm of uh, impulse generation. And this they were actually able to measure the frequency, and they said they saw that it is quite remarkable with that with those limited or those kind of equipments they were able to actually record to this level of like they were able to obtain this kind of information, and they saw that there is a frequency range from 5 to 100 impulses per second. Now Adrian try to understand why is this regular rhythm that is present, and he attributed this regular rhythm to the refractory period and the return of excitability of the muscle, sorry, of the impulse. So they are Adrian show basically was the one who came up with the idea that there is probably some refractory period that exists between different electrical impulses, and that is the reason for the rhythmic generation of the impulses. So that's why the correct answer to this question is the refractory period and return of excitability. Next, what scientific advancement in the 1950s revolutionized the study of biology? So, light microscopy was invented long before the 1950s. It was already existing, and like we discussed previously, light microscopy, the resolution of light microscopy was not sufficient to view the the organelles of the cell. So, no, the first option is not correct. The second option is the introduction of the electron microscope. Electron microscope was indeed introduced in the 1950s, and because of the use of electron microscope, scientists were able to actually understand the ultrastructure of the cells. Basically, they were able to look at the organelles of the cells, such as the Golgi, the mitochondria. All of these organelles they were able to visualize because of the introduction of the electron microscope. And later, of course, the synapses, even the suborganal structures, these were also uh, studied because of the introduction of the electron microscope. So the discovery of DNA revolutionized biology, yes, but that was more towards the I mean, it gave rise to the study of molecular. I mean, it contributed more to the study of molecular biology than to cell biology. Next, who introduced the term synapse in 1897? This we have already discussed. So it was Charles Sherrington who introduced the term synapse in 1897. Next, what key discovery about the chemical synapse was reported in 1954? So let me again go to the slides. So, yeah. So in 1954, during the American Association of Anatomists meeting, both George Pollard and Sanford's findings, as well as uh, Eduardo Roberta's findings, were presented. And at that time, the Eduardo de Roberta's presented the his work on the changes in synaptic vesicles. And basically, he observed the presence of synaptic vesicles in the nerves, and he observed that these. Then he came up with the idea that these synaptic vesicles probably contain neurotransmitters, and the fusion of these synaptic vesicles with the plasma membrane resulted in the release of these neurotransmitters, and that is how information is being transferred. The information that is present uh, in the neuronal cells is getting transferred. So this was actually the key discovery about the chemical synapse that was reported in the year 1954. Basically, the visualization of synaptic vesicles and their role in neurotransmitter release. Although initially the role of the synaptic vesicles in neurotransmitter release was just an idea that de Robertis proposed, but later it was actually shown that it is indeed true that these synaptic vesicles indeed contain neurotransmitters, and these neurotransmitters are released upon vesicle fusion, and the released neurotransmitters then go and bind to the receptors, their respective receptors on the postsynaptic membrane. Next, what organism did Hodgkin and Huxley use to study action potentials? So this also we had discussed. Hodgkin and Huxley used the squid uh, as a model because the squid has a giant axon which allows easy recordings and other recordings of action potential. Next, which neurotransmitter did René Cotiu focus on in his research at the neuromuscular junction? So, yeah. So René Cotiu was a French researcher. So initially he had studied medicine. Later he became interested in research, and he's decided to do his thesis to understand how the neuromuscular what is the neuromuscular junction like, and he because at that time there was a debate regarding neuronism versus reticularism. He was also caught up in that, so he also wanted to understand whether the the neuron doctrine is true, or whether the the neurons are actually just a reticular network. Basically, whether the neuron doctrine is true or the reticular theory is true. So, in order to understand that, he decided that he will use the NMJ as a model. So NMJ basically is the neuromuscular junction. So he decided that that it will be a good idea to use the neuromuscular junction as a model. But the problem was that the methods that were available to him at that time, they were not sufficient. So at that time, there was a limitation because there weren't any fluorescent staining methods and so on that were available. It was in the 1930, 1910s, 1920s, 1930s, that time. So naturally, there was a limitation that he had to deal with. And what was the limitation? The limitation was that there were only two ways of staining tissues, staining the nervous tissue. That either it was using gold or silver, or it was just vital staining with methylene blue. Neither of these methods provided any clear visualization of the neural and muscle membranes at the microscopic level. So it was not possible to identify whether the neuromuscular junction was actually, I mean, whether the structures of the NMJ were actually separate, that is, the neuronal doctrine was true, or whether they were fused or continuous, that is, whether the reticular theory was true. So, what Rene did was that at the time in the mid 1930s, it was established that acetylcholine is a neurotransmitter. So, acetylcholine, being a neurotransmitter, means that there must be something that, so once the action of acetylcholine on the postsynaptic membrane is over, there must be something that inactivates the acetylcholine. So this people who at the time were speculating that there is an enzyme called acetylcholine esterase that can inactivate the acetylcholine after it has acted on the postsynaptic membrane. So he decided to investigate the role and localization of acetylcholine esterase. So, in collaboration with David Nachmanson, who was a German Jew who had to flee from Nazi Germany at that time, uh, Rene and David together developed biochemical methods to study this acetylcholine esterase at the neuromuscular junction. And what they discovered was that this acetylcholine esterase is located at the sites where the nerves innervate the muscles. Basically, it is located at the neuromuscular junction. And in fact, it is able to persist even after the nerve regenerated. Basically, that this suggested that the acetylcholine esterase was present in the junctional membranes themselves. And what Cotu did was he developed this Janus green bead dye staining technique and used that to reveal the presence of synaptic gutters and subneural apparatus at the NMJ. So synaptic gutter is basically the synaptic cleft, whereas the subneural apparatus is basically the postsynaptic side. Let me just go to the presentation. Yeah. So this is the Janus B staining that Rene Cotiu and David Nachmanson did, and this reveals the presence of the acetylcholine esterase at the sites where the nerves innervate the muscles. And basically, you can see the presence of these round concave structures that basically shows that there are presence of synaptic gutters and the presence of a lamellar subneural apparatus on the. So basically, you can see that these are highly lamellar structures. So he called them subneural apparatus. So Rene Cotiu's work strongly supported the neuronal doctrine, and basically he used an NMJ to show that the cells are indeed separated by synapses and they are not continuous reticular networks. Now coming back to this question, which neurotransmitter did Rene Cotiu focus on in his research at the neuromuscular junction? So we now know that he focused on acetylcholine. Basically, he worked on the enzyme acetylcholine esterase, which inactivates the acetylcholine post its release at the neuromuscular junction. Next, what principle explains the rapid changes in ion flow during an action potential according to Hodgkin and Huxley? Okay. Synaptic vesicle theory. That has nothing to do with Hodgkin and Huxley's uh, work because that was the presence of synaptic vesicles and the concept of vesicle fusion. These were all carried out by D. Robertis, as we discussed, and several other people, of course. So the synaptic vesicle theory, it does not have anything to do with the Hodgkin and Huxley model. Sliding filament hypothesis is for the actin and myosin based muscle, like the structure of the muscle sarcomeres. That is the sliding filament hypothesis. Genetic information flow again has nothing to do with the Hodgkin and Huxley model, because that is something that we discussed previously, right? We have discussed the flow of genetic information from DNA to RNA to protein. That is completely different from the uh, iron flow hypothesis that was proposed by Hodgkin and Huxley. So what did Hodgkin and Huxley uh, what did they show? They show that there is flow of sodium and potassium ions across the membrane, which is responsible for the action potential. So the flow of sodium and potassium ions is the uh, is what is responsible for the changes in changes responsible for the changes in the potential of the membrane, which results in the action potential. Let me just go to the text and discuss it a little more. Yeah. So, the iron flow hypothesis of Hodgkin and Huxley basically proposed that the action potential results from selective flow of sodium and potassium ions across the membrane. And how does the selective flow happen? The selective flow happens because of the presence of ion channels in the plasma membrane. So basically, they say that the using the ion flow model, they explained how differences in the relative concentrations of sodium and potassium and across the plasma membrane results in the electrical changes that are observed during the action potential. I hope that is clear to you. Next. What did Goldman-Hodgkin-Katz equation formalize? So we had already discussed this, that the Goldman-Hodgkin-Katz equation formalized the membrane potential differences from ion permeability and gradients. So the Goldman-Hodgkin-Katz equation basically formalized the how the membrane potential arises because of the relative concentrations of each of potassium, sodium chloride, as well as the permeability of these ions across the plasma membrane. Remember, the permeability of these ions arises, so the differences in the permeability of these ions arises from the differences in the permeability of the ion channels that are present in the plasma membrane. So the ion channels for these different ions allow basically, excuse me, so the ion channels that are present in the plasma membrane basically mediate the flow of these ions across the membrane. And therefore, the relative permeability of the plasma membrane depends on the types of ion channels that are present and the permeability of those ion channels to each of these different ions. Moreover, the concentrations of these ions in the interior as and in the exterior of in the interior of the cell and outside are different, and because of the differences in the concentration there is a concentration gradient. So there is it means that because of the presence of the concentration gradient, uh, when the ion channels are open, the uh, the potassium, sodium, and chloride ions move on the basis of their concentration gradient from a higher concentration to a so from a net higher concentration to a net lower concentration. But also, we need to remember that because of the because of the presence of sodium potassium pumps and the presence of ion channels is the reason why there is a membrane potential that arises. So the Goldman-Hodgkin-Cards equation formalized the net potential differences from ion permeability and gradients. Next, which method introduced by Kenneth Cole was critical for studying nerve excitability? Okay, just a minute. Okay. Um, so Kenneth Cole was the person who decided to host Hodgkin and Huxley. So he invited them to work with him. Just a minute. Uh, yeah. So Kenneth Cole invited Hodgkin and Huxley to work with him to understand the electrical properties of cells and their membranes. So he is the one who introduced the use of the voltage clamp technique. So, which actually revolutionized the study of ion channels, and the voltage clamp technique is something that is used till date in electrophysiology and continues to provide us with insights into how neuronal cells conduct uh, electrical impulses. So, Kenneth Cole introduced the use of the voltage clamp technique. So the correct answer for this question is the first option, voltage clamp technique. Next, what was a key finding of Bernard Katz's 1939 monograph on nerve excitation? Again, let me go to the text. Yes. So basically, Bernard Katz monograph, The Electrical Excitation of Nerve, compiled both his doctoral work and subsequent studies, and it offered a comprehensive overview of nerve excitation mechanisms. So the key finding of this monograph on electrical excitation of the nerve basically basically allowed us to basically provided an understanding of the electrical properties of nerves. What technological breakthrough allowed Hodgkin and Huxley to measure action potentials accurately? So the correct answer to this question is the cathode ray oscilloscope. Although, right, I mean, at that time, the cathode ray oscilloscope was actually regarded to be a very technologically advanced equipment. Although now we do not regard it and the same, but this the use of the cathode ray oscilloscope was extremely useful for Hodgkin and Huxley to actually carry out action potential measurements with accuracy. So again, this is so the cathode ray oscilloscope, which was invented by Herbert Gasser and Joseph Erlanger in 1922, allowed the uh, recording of nerve impulses. And this cathode ray oscilloscope was later used by Hodgkin and Huxley. Just a minute. For their experiments with the squid giant axon, they used the cathode ray oscilloscopes and electronic amplifiers, and that allowed them to actually measure the nerve activity with great accuracy. So with this, I come to an end of the previous year assignment questions. I hope you were able to understand the understand so far. And now I'll start with discussing some of the concepts that were covered in this week's session. And in case you have any doubts, we can discuss that afterwards. So, like I had started telling you, Ramoni Kahal was the one who first proposed the neuronal doctrine, the neuron doctrine. So, while he was making observations of the mammalian cerebellum, he observed that these structures are discrete. They are not a continuous reticulum as some people were proposing. So, therefore, Kahal said that the nerve cells are independent elements, and the propagation of nervous action between these nerve cells is made by contacts. So, he proposed that there might be some contacts. Much at that point, there was still a lot of debate regarding uh, Kahal's point of view, because Golgi and several others proposing the reticular theory. Basically, that the nervous system is a continuous reticulum rather than independent elements, is what the reticular theory was proposing. So, Sherrington introduced the term synapse in 1897 while he was studying the reflex action in the spinal cord. But still, many people were skeptical about whether there is indeed discrete neuronal cells or it is a reticulum or not. So at that time, Willy Kern, who was a German investigator, he worked extensively on the neuromuscular junction in the 1860s and 1870s. So he studied the neuromuscular junction extensively, and he suggested that at least at the neuromuscular junction, the impulse that is coming from the axon, basically the impulse that is propagating along the axon into the terminal, leads to the activation of the muscle contraction by some unknown mechanism. And he suggested, so this was already known that there is some mechanism by which the impulse from the axon is stimulating muscle contraction. But Kern suggested that the whatever is happening at the NMJ, it could be basically the this discontinuous the mechanism that This impulse is coming to the axon terminal, and it is somehow propagating to the muscle and resulting in the contraction of the muscle fiber. A similar thing could be happening to all for all the connections that are in the nervous system. So basically the connections between nerve cells could also be similar, that the nerve cells might be discrete entities and there is some sort of impulse propagation between the nerve cells that is similar to what is happening at the neuromuscular junction. uh, Emile Dubois Raymond, who was a German physiologist, he had suggested in 1877 that this muscle activation might somehow involve some stimulatory secretion by the nerve endings. So basically, he said that there is probably something that is getting secreted from these nerve cells that is being detected by the muscle, and because of that, the muscle is contracting. But uh, this suggestion of by Emile Dubois Raymond was largely ignored. So then came René Cortu. So René Cortu was born in 1910, if I'm not wrong, and he was a French researcher. And René Cortu, because he published most of his work in French, many of his work did not. Was not as widely known as several other researchers because, again, like we know, publications in English are more widely people more they are more widely received and people are more aware of those publications rather than those that happen in other languages like in French. So publications in English, English is still the language in which most publications happen even now, and publications in other languages do not reach a wider audience like those that happen in English too. So Rene was a French researcher who, like we discussed a little while ago, used the neuromuscular junction to address the issue of whether it is a whether the nervous system is composed of individual neuronal cells, that is the neuron that are separate entities, that is the neuronal doctrine, or the reticular theory. And we had discussed the limitations of the staining techniques and also the limitations imposed by the light microscopy that was that only which only that was available at that time, and that also imposed limitations. But despite that, Rene Cotiot was able to make his observations of the presence of synaptic gutters, which basically provided the first support to, which provided the strongest support to the neuronal doctrine. And he did this with the use of the Janus green B dye. During around that time, George Cole developed a histochemical method to localize acetylcholinesterase by staining for a closely related molecule to acetylcholine. So he stained for acetylthiocholine instead. And this allowed more easy visualization of the acetylcholinesterase. So this was called the Coel method. And Coel method was then widely adopted by other people to demonstrate the cholinergic systems that were present in the nervous system. This coil method was also adopted by Cotu and Taxi. So they made an improvement to the coil method, and using this improved coil method, they again confirmed what Cotu had shown with the Janus green B dye. And remember all of this that Rene did, he was doing during the tumultuous times of the World War II. It was a time of war that was happening, and despite that, Rene Couture actually made such significant contributions. He managed to carry out such experiments, and those have now those have laid the groundwork for understanding synaptic structures and functions. Remember at the time, even without electron microscopy, those people managed to uncover the presence of synapses. We have also discussed the chemical synapse briefly when we were talking about work that was done by Sanford Paley and George Ballard. So, Paley and Ballard had collaborated with each other and they used electron microscopy to understand the different structures that were present in the neuronal cells. At the same time, Eduardo de Robertis and Bennett, these two also worked to understand, these two are also using electron microscopy and to understand the synaptic vesicles, to understand the neuronal ultrastructure and so on. So Sanford Paley, uh, was initially a student of Ernst and Berta Schrader. So these were European scientists. So he was a young student who was working with them, and he has described in his memoir how his time with these two scientists opened his eyes not just in terms of science but also in terms of culture, in terms of his exposure to the European style of sophistication and so on. And when he returned back to America, he was working at University of at Yale University basically, and he had to juggle both teaching responsibilities as well as his newfound interest in using electron microscopy. So at that time, he collaborated with George Ballard, who was, like I said, who was a refugee from Romania because at the time war was happening. So he had to leave, he had to run away from Romania and he sought refuge in the US. And both of these people, they collaborated to use the electron microscope to understand the the final structures of the nervous system. So at that time when Sanford Paley and George Ballard collaborated. There were also advances in the staining techniques and the tissue sectioning techniques that were taking place. So they decided to use these as well as the electron microscopy and find out the ultrastructure of the neuronal cells. So at that time, the Sanford basically made the following discoveries. He identified the presence of organelles such as the endoplasmic reticulum, the presence of neurofilaments, Golgi apparatus, and missile substance in the within the neurons. Moreover, he also was the first to take EM images of the synapse, which he did by studying the motor cells of abducens nucleus. So he was the first one to report EM images of synapse in 1954. Just a minute. Okay. Oh, sorry. Yeah. So, Sanford Pele, George Ballard, Eduardo de Robertis, these people were all contemporaries, which means that they were all working on, uh, they were in the same field at the same time. So Eduardo de Robertis, again, he was also trained in medicine, and then he started showing interest in biological research, and he started focusing on research. So, together with Frank Schmidt at MIT, his early EM studies of axons showed that there are neurotubules that are present within these axons. And later he went on to establish an EM facility in South America in Uruguay. And he performed several seminal studies on the retina's fine structures. And in collaboration with Bennett in Seattle, he also investigated synaptic membranes and exocytosis in adrenal medulla cells. So he also observed the presence of synaptic vesicles in neurons. So at that time, there were so both Sanford Pele and Eduardo de Robotis claimed to have been the first to visualize the synaptic vesicles. So there was both of them claimed that they were the ones to have first observed and first documented the presence of such synaptic vesicles. I hope it is clear to you so far. Uh, okay. Let us come to, let us briefly, I think we have already covered this, but we'll still just briefly discuss some of the findings um, that were discussed by Sanford Pillay and George Ballard at the American Association of Anatomists. So, both Sanford Pillay as well as George Ballard, they presented their work at this American Association of Anatomists in 1954. So, George Ballard uh, presented his work on the EM observations of interneuronal and neuromuscular synapses. And he observed the presence of mitochondria, vesicles, as well as dense plasma membranes at the contact points. And he also showed that there is similarity of the neuromuscular synapses at this that that are present over the rat skeletal muscles with those that are with the central synapses that are present in the brain. Then Sanford Paley's a report in which was assisted by George Ballard. He presented his work on the EM observations of the cytoplasm of neurons, and he discussed his findings of the Nissl substances and the Golgi body and dendrites and sorry, and Golgi body. So remember, earlier Golgi body was shown to be I mean, was dismissed by some people as being just an artifact of staining. But later, Sanford Pillay actually showed that Golgi bodies indeed, I mean, he observed that Golgi bodies are also present in the neuronal cells. Eduardo de Robertis' findings were the changes in synaptic vesicles. So he used guinea pig cochlea and he tried to look at the synapses, and he also described the presence of synaptic vesicles, mitochondria, and ER at the presynaptic endings. So yeah, there is one thing that is important which I want to tell you here. So Paley's 1956 paper, in which he defined a few key features of synapses, these were that one, uh, the presynaptic and postsynaptic membranes were closely limited, means that they were very close to each other. I mean, they should be very close to each other. Next, the presence of synaptic mitochondria. The third, the presence of synaptic vesicles. And fourth, the absence of protoplasmic continuity. Basically, saying that the reticular theory is not correct because uh, in synapses, the two different independent nerve cells are contacting each other without the presence of any protoplasmic continuity. So, yeah, so this we were discussing a little while ago, right? That both Sanford Paley and D. Roberts and Bennett said that they each were the first to observe synaptic vesicles. So they were both focused on asserting they are them being the first to observe synaptic vesicles, and while they were both acclaimed researchers, they both each claimed that they were the first to observe the synaptic vesicles. Next, uh, so George Gray in London, he when he was looking at these synapses, he tried to differentiate between two different types of synapses. So he said that there are type 1 synapses and type 2 synapses. And type 1 synapses, according to him, were excitatory synapses, whereas type 2 synapses were inhibitory synapses. And the type 1 or the excitatory synapses were asymmetrical in nature, whereas type 2 or inhibitory synapses were symmetrical in nature. Do you have any questions so far?
No, ma'am.
Okay. So, we also had discussed the gap junction briefly. So, let me just recap what are gap junctions. Gap junctions are basically structural contacts between individual neuronal cells. So, gap junctions exist not just in the nervous system but also in several other tissues. But the gap junctions in neuronal cells are responsible for electrical transmission of the of the impulses. Basically, they form the basis of electrical synapses. So these electrical synapses were initially thought to be present only in invertebrate systems, but later they have also been found in vertebrate brain stems. And these gap junctions were discovered by Rebel and Karnowski in 1967, and they were shown to be the structural basis of these electrical synapses. Gap junctions facilitate electrical conduction and molecular diffusion between cells. So basically, these are made of complexes of proteins that interact with each other, which allows the exchange of nutrients as well as informations and molecules, and also electrical signal transfer. And their functions in other parts of the body are also similar. They also, like I said, are present in not just in the nervous system but also in some other parts of the body. And they're also they play a role in allowing the exchange of information between different individual independent cells. So the discovery of gap junction does not invalidate or does not disprove the neuron doctrine. So even if there are gap junctions, the gap junctions are simply a form of direct communication. They are the cells are still individual and they still have their own functional autonomy. So I hope you have understood what gap junctions are, and I hope I have been able to explain the neuron doctrine to a certain extent. We will come back to the neuron doctrine again in later lecture videos, and we will be discussing the neuron doctrine and the reticular theory in even more detail. So, let us discuss about Betty Guerin's discovery of the myelin sheath. So, myelin is basically a fatty substance that surrounds the nerve fibers. So as you can see, this is the axon, and the myelin sheath basically wraps around the axon. It's a multi-layer wrapping of the axon, and this myelination. So basically, myelin is a fatty substance, but it is actually because of the glial cells. So Schwann cells are a type of glial cells that are that basically uh, he- help in the formation of this myelin sheath around the axons. And this multi-layer wrapping of myelin around the axons basically helps in the conduction, faster conduction of nerve impulses, basically. So the rapid action potential conduction is aided by the presence of this myelin sheath. So we will come to saltatory conduction in I think in future slides. We will be talking about future lecture videos. We'll be talking about what saltatory conduction is. So myelin sheath basically, because of their high resistance and low capacitance, they help in saltatory conduction or rapid action potential conduction. And there are several diseases in which demyelination of axons results in disorders like multiple sclerosis. So the presence of myelin sheath is actually very important for rapid action potential conduction. Interestingly, there are also axons which are not myelinated. For example, in organisms like fruit fly, Drosophila melanogaster, the myelin is not present, so axons are not myelinated. Whereas in the human brain, there are myelinated axons that are present. So I hope so far uh, whatever I have explained is clear. I would really recommend that you go through some of these articles. These provide excellent reviews of whatever we have discussed so far. For example, the image of the synaptic gutter that I had shown, that was from this paper by Suji. So that describes the work of Rene Cotiu, and the fact that he did not I mean he's not as although he was the first histologist to introduce the term synapse, the relatively less recognition of his contribution, so that is has been highlighted by Suji, and the work of Sherrington, for example, although we have discussed only about the fact that he coined the term synapse, Sherrington has made several other discoveries for which he received the Nobel Prize in Medicine. So this paper is also extremely interesting because it details the work of Sherrington not just in the nervous system but also in other fields such as understanding of cholera or the development of the diphtheria vaccine and so on. So it tells us that scientists at that time were very creative. They were also they also had several ideas that they were not afraid to test, and they made use of the limited equipment that was available to them, but still managed to come up with remarkable discoveries. So I would really recommend that you go through some of these papers, and it will really give you insights about how the kind of process the people at that time had, and despite whether I mean many of the things that they have done have actually laid the foundations of the current modern neuroscience as it is. So I would really recommend for you to go through these papers. Next. So, we'll just quickly go through some of the other. Just a minute, Soumya. I think you have posted something in the chat box. Are the nerve conductions in Drosophila slower than in humans as they lack myelin? Uh, I'm not aware of that. I think it is something that I'll have to look up. So maybe I can check this and I can get back to you. Uh, I can maybe send you a reply in the discussion forum. Would you? Okay. I need to check whether the nerve conductions in Drosophila are different. I would expect that because of the lack of their myelin sheath, it would be different. It would be reduced, is what I expect, but I would have to actually go back and check whether it is indeed the case. Okay. Uh, this is also something. Okay, no. Okay. Now coming to nerve impulses. So far we had been discussing about synapses. Now let us discuss about the how nerve impulses were studied, and how people have like the seminal work that was done by Hodgkin and Huxley, for example, which is like the lore of neuroscience. We'll slowly come to that. I hope we have enough time to discuss that. Let us start with some of the major discoveries that were made with regard to nerve impulses. So in 1791, Luigi Galvani discovered animal electricity in frog muscles. So basically, he observed that frog muscles were twitching um, in response to. So basically, he observed that frog muscles they have some electrical component in them, basically because of which they undergo twitching. Then in 1819, Hans Christian Oersted discovered the electromagnetic effect. In 1840, Carlo Matteucci and Emile Dubois-Raymond used galvanometer in electrophysiology. So this is the same Emile Dubois-Raymond who had proposed the idea that there might be something that is being secreted at the neuromuscular junction. But then his idea was ignored at that time. Then, in the late 19th century, German physiologists took an oath that they will try to understand the mechanistic physiology. Basically, what is the underlying, what is happening in the level of mechanisms in the nervous system. In 1888, Walter Nernst developed the Nernst equation. In 1902, Julius Bernstein uh, basically came up with theories on the resting membrane potential and the action potential. So, the resting membrane potential is basically the difference in the membrane potential between the interior of the cell and its surrounding environment. Basically, the between the interior of the cell and the extracellular environment. And generally, the membrane potential of most cells is negative. So it is close to around minus 55 to minus 60 volts. Then in the early 20th century, uh, action potentials were studied in various organisms. For example, the use of the squid giant axon, for example. And then by mid 20th century, people actually try to understand how these action potentials are propagating. So, we need to understand that at that time there were a lot of limitations. It was not easy to record nerve impulses because now, even with the use of extremely sophisticated equipment, we still do have several challenges to record nerve impulses. Imagine in the 19th century when the equipment was far more primitive compared to what we currently employ. It was very difficult to record nerve impulses with accuracy because the instruments were not as sensitive as the ones we use currently. So during that time, the advent of the galvanometer was a very important breakthrough because it allowed indirect measurements of nerve activity. Although it did have limitations in speed and susceptibility to vibrations, it still allowed some extent of measurement of nerve activity. This also we had discussed that Keith Lucas was the one who propelled the era of the electromechanical era, basically. But Keith Lucas died in an accident, and his student Edgar Adrian took over the work, and he made several important contributions in the early 20th century. So remember, these were extracellular recordings that were being made. They did not have the sophisticated equipment to make intracellular recordings. So what Edgar Adrian did was he studied the efferent impulses, basically the ones that are uh, the sensory impulses basically. So he studied the efferent impulses from receptors in the frog's sternocutaneous muscles. And how did he study that? He used capillary electrometer and a three-stage amplifier. So and the stimulus that he applied was stretched the muscles with varying weights, and he wanted to record whether stretching the muscles with different weights was changing the nerve impulses that were being propagated. And what he found was so his key findings were that uh, there is a regular rhythm by which the impulses are being generated. And he managed to record that there is the frequency of this impulse generation is between 5 to 100 impulses per second. And he attributed this rhythmic generation of impulse to the refractory period and the return of excitability. So he said that basically there is impulse generation, then there is a refractory period, then again the excitability returns. That is the reason why there is a rhythmic rhythm. There is a regular rhythm by which the impulses are being generated. So he also uh, he also came up with the all or nothing response from his findings. So the all or nothing response is that he observed that the electrical response in the nerve fibers shows an all or nothing relation to stimulus strain. Means that either there is impulse or there is no impulse. There is no in between, basically. Next, we discussed the uh, cathode ray oscilloscope breakthrough, which was a very important innovation because it allowed the later experiments. It basically aided the later experiments that were carried by Hodgkin and Huxley. So the cathode ray oscilloscope was a major breakthrough that happened, and this oscilloscope could be used to record nerve impulses, and it revealed both the duration, also revealed the duration of the impulses, which is basically a few milliseconds of time. And this was actually a major neurophysiological discovery. So at that time, there were certain people who called themselves, certain scientists who called themselves as axonologists, and they tried to come up with theories of nerve excitation. So amongst these people were Henry Lappé, Hill, Rushton, Rashevsky, and Monnier. So the all of these people gave with different types of theoretical contributions to the field. So their idea was to say they wanted to come up with some physical explanation for the action potential. So, in addition to the theories that these axonologists proposed, there was also technical innovation that happened. So, The introduction of micropipettes was a very key technological innovation that happened, and these micropipettes were initially used to record from large plant cells. And these micropipettes were made by using microforges. So again, microforges were another innovation. These allowed the creation of extremely precise micropipettes, which allowed intracellular recordings in plant cells. So Nikolai Rashevsky, so he is one of the persons uh, whose theoretical contributions at that time towards nerve excitation was that he proposed mathematical models for nerve activity. Then Henry Lapide introduced concepts of Rio base and Chronaxy to describe nerve excitation. So these like some of these theories at that time, they were basically trying to come up with physical explanations rather than chemical explanations. So basically, Rio base was for the strength of the impulse, and Chronaxy was for the duration of the impulse. Then Archibald Vivian Hills or A.V. Hill, was the one in whose lab Bernard Katz spent his. Basically, Bernard Katz was again a refugee who spent his time in Archibald Vivian Hill's lab. So Archibald Vivian Hill made significant contributions to the understanding of the physics of muscle contraction as well as nerve impulses. Then Henry Lapide tried to formulate universal laws of nerve excitation using his concepts of Riobase and Chronaxy, and he tried to establish universal laws to bridge both physical science and neurophysiology. So all of these people, Rashevsky, Monnier, Hill, Rushton, and Lilly, and several others also, all of these tried to explore this electrical excitation of nerves through theoretical and through physical models. So then came Bernard Katz, and as a student, Bernard Katz wrote a monograph called Electrical Excitation of the Nerve, and this publication was actually a very significant contribution because it tried to synthesize that during that period of time, it tried to synthesize whatever was that period's current knowledge on the electrical properties of the nerve and tried to basically to understand how nerve impulses work. Bernard Katz's monograph was a very important piece of work. And like we discussed, at that time, advances in micro, sorry, the advances in using micropipettes for intracellular recordings had just started, and people were using initially using the plant cells Nutella for this purpose, and they showed that these electrical phenomena, these action potentials, are universal. They are present in all forms of life. They are even present in these Nutella cells. Finally, let us just briefly discuss the Hodgkin and Huxley legacy, in which they unraveled how the action potential propagates. So both Hodgkin and Huxley were extremely, they were very brilliant people who managed to decode the mysteries of the action potential. So Hodgkin was so these both of these people, they were fortunate enough to meet with very great minds of that time, such as J.J. Thomson and Ernst Rutherford and so on. So not just were they pioneering minds, they were also able to interact with such other pioneers, which of course propelled their own scientific mindset and also allowed them to make the contributions that they have made. So, it's important to understand that both Hodgkin and Huxley, because of their prior endeavors, so their background in, for example, Huxley's background in physics and Hodgkin's experiments on local circuits and graded potentials, all of this background further enabled them to uh, make the breakthrough that they did with the squid giant axon experiments. And the reason that they chose the squid giant axon was because of its size. So it is a very large axon. It makes the recordings much easier to do. And what these experiments with the squid giant axons revealed was the dynamic changes in membrane potential. So we had discussed this previously also, that the changes in membrane potential because of the uh, movement of the sodium and the potassium ions across the membrane is what actually constitutes the action potential. So these changes are extremely dynamic. They happen over very small time scales, and this is the reason why action potential is the propagates along the axon. So again, at that time, it was World War II was happening, and both of these scientists had to was pause their research during that time, and then post-war they again resumed their research, and now they had a lot of war surplus electronic equipment, which was of course a good thing for them. Uh, and because of the fact that the war was over, they also had the right kind of equipment with them, and they now were able to again focus on studying the ion movements across the nerve membranes. This allowed them to understand the how the action potential is propagating. So they are using their observations and their recordings in using the squid giant axon. Hodgkin and Huxley proposed the ion flow hypothesis. So what did they propose? They proposed that the action potential results from the selective flow of sodium and potassium ions across the nerve membrane. So the flow of these is selective, it is not random, and this happens across the nerve membrane, basically between the intracellular and the extracellular uh, compartments of, basically the intracellular compartment of the nerve cell, I mean the neuronal cell, and the extracellular environment. And this hypothesis that they proposed, they also had a lot of experimental evidence to support the hypothesis. It is very important that when we propose a hypothesis, that also has to be followed by rigorous experiments, because hypotheses are just our ideas that we are proposing based on existing literature. But unless those hypotheses are actually tested by rigorous experimental evidence, we cannot blindly accept those hypotheses. All hypotheses have to be rigorously tested. So, Hodgkin and Huxley's rigorous experiments and the evidence that they uncovered, the results that they uncovered, which were strongly supported the hypothesis that sodium and potassium channels are moving selectively across the neuronal membrane, and that is how the action potential is propagating. And their iron flow model explained how the differences in the iron concentration across the membrane led to the rapid electrical changes that were observed. So the because there was difference in the iron concentration across the membrane, that is the reason why the sodium and potassium ions were able to uh, flow across the membrane. If there is no difference in ion concentration across the membrane, then the uh, sodium and potassium ions will not tend to move across the membrane. So there must be a concentration gradient of both the sodium as well as the potassium ions across the membrane in order for the flow of these ions across the membrane. So the Hodgkin-Huxley model has had a very, very profound impact on neuroscience because it provided the foundation on how nerves actually communicate. How is the information being passed along the nerve itself? So that's why these experiments that were carried out by Hodgkin and Huxley are extremely seminal in nature, and they have influenced all the they have influenced several other studies that basically they have influenced our understanding of how nerve impulse conduction actually takes place. This also we had discussed that Kenneth Cole was one more pioneer in the field of neurophysiology because of his introduction of the use of the voltage clamp technique. Basically, the voltage is like kept more or less the clamped or relatively constant so that the using a recording electrode and a stimulating electrode the intracellular recordings can be carried out. So Kenneth Cole had collaborated with Hodgkin and Huxley, and he had provided several crucial insights as well as the technological advancements that facilitated Hodgkin and Huxley's groundbreaking research. I think we have already discussed Goldman-Hodgkin-Katz equation and its importance to the importance in terms of formalizing the membrane potential in because it basically tells us how to determine the how to understand the membrane potential in terms of both the permeability as well as the concentration gradients of these ions, and basically takes into basically explains the reversal of the potential across the membrane, and it gives us a model of how the uh, it basically gives us a model by which we can uh, understand how the membrane potential is arising. I think this we have already discussed to different extents, so I will not rehash this again. I would recommend you to, I would strongly recommend you to read Hodgkin and Huxley's articles in which they have performed the squid giant axon experiments, and also these other review articles in which they explain how Hodgkin and Huxley carried out their experiments and what is this is the Hodgkin and Huxley article. I would also recommend that you go and read these articles and understand how Hodgkin and Huxley carried out the experiments using the squid giant axon, and how they, what they uncovered, and how they understood how the action potential is propagating. So with this, I come to the end of today's live interaction. I hope uh, I hope you were able to understand the concepts from this week. In case you have any questions or any doubts, feel free to post them in the discussion group. I would be happy to reply to you. So, thank you very much. With this, I will stop the meeting. Do you have any questions?
No, ma'am.
Okay, thank you very much.